Good morning. My name's David Bacsik, and this year I am a postdoc working with Dr. Pablo Sukiyama at Universidad Peruana Cayetano Heredia in Lima. But before this, I worked with Dr. Jesse Bloom at University of Washington to develop single-cell methods to study influenza virus. And when we started sequencing individual cells infected with influenza, one of the first things we realized was that the variation in how much viral gene expression occurred in each cell was extreme. In some cells, or in most cells, there's very little viral gene expression, maybe 1%, 5% of the transcripts come from virus. But in some rare cells, more than half the viral transcripts come from virus. So there's actually more viral material expressed than host material. As we were trying to explain this difference between cells, we thought maybe viral mutations were playing a role because influenza virus has a very high mutation rate with almost no proofreading. We quickly ran into a problem with standard single-cell sequencing methods. As many of you know, single-cell sequencing counts the number of transcripts in each cell, but it doesn't tell you much about the sequence of those transcripts. This is important because many biological systems have significant genetic variation between cells. Influenza virus is one example, but cancer genomes are another. And so to address this problem, we developed new methods to enrich specific transcripts that we were most interested in and use long-read sequencing to capture their complete sequence. Today, I'm going to tell you a bit about these techniques and how we applied them to study influenza virus, but I want to emphasize that the same approach could be used to enrich any target that you are interested in. So in our single-cell sequencing libraries, we had a mix of viral transcripts and host transcripts. And overall, the viral transcripts only represented approximately 1% to 2% of the total number. And so we needed a way to select just the viral transcripts for long-read sequencing. The solution that we came up with was circularization, and there were two important goals that we were trying to meet. First, we needed a way to amplify a specific target, in our case, the viral genes, and not the host genes. And second, we needed a way to retain the cell barcode, so we know which cell each transcript and each sequence comes from. We started with standard single-cell RNA sequencing library, shown here, in box 1. So this is the full-length transcript before fragmentation. And at this point, there is a gene sequence shown here in green. There is a cell barcode shown here in purple. And there are two adapters, one on the left side in gray and one on the right side in blue. And the first step in this method is to use ligation or PCR or bridge oligos, whatever molecular biology you prefer, to replace the adapter on the left-hand side with a duplicate sequence of the one on the right-hand side. Once you have these homologous sequences, you can do a sequence-specific ligation reaction where these two sequences bind and the molecule is ligated to close and form a circular molecule. At this point, you have a circular version of your barcoded transcript, which has no beginning and no end, and you can use specific primers to amplify just the transcripts that you are interested in. These primers bind in the transcript sequence, and you produce a linear molecule, shown here in 4, that splits the transcript sequence you're interested in into 2 parts, and you have the cell barcode retained in the middle. From here, you can use standard long-read sequencing technology, whether that's PacBio or Oxford Nanopore, to generate these complete sequences. And then the bioinformatic pipeline is basically standard. There are only 2 additional steps. First, you need a reference genome that looks like this molecule in number 4 that you can align your reads to. And then after you generate a consensus sequence for that molecule, you need to basically copy and paste the sequence of the 2 halves and put them back together in the correct orientation. Once you do that, you have data that looks like this. These are consensus sequences for the influenza virus genome that infected each cell in our data set. In this plot, each row represents an individual cell that was infected with influenza virus. Influenza has 8 genes on 8 different segments. You can think of the segments like the virus's chromosomes, basically. And each segment is represented here by a blue arrow. There's very little non-coding sequence in the influenza genome, so we're missing 50, maybe 100 base pairs at either end, and we have something like 90% of the complete genome for each cell. The first cell that I'm showing here is infected with a completely wild-type genome without any mutations. So each of these bars is totally blue. But this type of infection is actually very, very rare. Much more common is to have at least one SNP, and SNPs can be synonymous, shown here in green, or they can change the protein coding sequence, shown here in orange in the third cell. They can have deletions, which are shown here in pink. The third cell down has a small deletion, and the fourth cell down, circled, has a very large deletion, removing almost the entire protein coding portion of that gene. Because of the way the influenza virus replicates, these types of large internal deletions are very common, and they essentially knock out the function of that protein. The last type of genomic mutation that we see in influenza virus is the complete absence of a segment. So here, this last cell has 7 of the influenza segments, and they're all wild type, but it's completely missing the last segment. That last segment is called NS, and I want to highlight this case specifically because this turns out to explain why some cells make massive amounts of influenza virus transcripts. If we look at this data tied back to the gene expression, and we look at the fraction of all mRNA transcripts in each cell, shown here on the x-axis, you can see that cells that are missing this NS gene, either through segment absence or massive deletions, express much higher fractions of mRNA on average, and there are several of these very, very productive cells where more than half of the viral transcripts, more than half of the transcripts in the cell come from virus rather than from host. This is a big contrast to cells that express all 8 viral genes, or cells that are missing other segments besides NS. And it turns out the reason for this is that NS plays an important role in exporting viral genomes from the nucleus and starting the process of releasing new viruses from the cell. So basically, cells that are missing the NS gene have a pathologic accumulation of viral transcripts in the nucleus, and this is totally different than what I expected. I expected the viruses with the complete normal genomes to be the best at transcribing, and that's not the case at all. So this work was very collaborative. Several people in the Bloom Lab contributed, and I want to highlight specifically Bernadetta, who is another postdoc in the Bloom Lab, and Alistair Russell, who worked really hard to get the technical details of this approach right. And in sum, I want to highlight that this method of circularizing your transcripts from a single cell sequencing library can be used to target any specific transcript that you want to know the sequence of, while still getting this single cell gene expression information. In our case, we found that influenza virus genetics vary significantly from cell to cell. This was not surprising, but it hadn't been shown before. And we found that loss of this specific influenza gene, NS, results in abnormal high virus transcription. And high viral transcription does not necessarily represent success of the virus in infecting the cell, but rather represents an abnormal dead-end pathway. So this work was published in eLife, and you're free to read up on the methods. We ended up tying this not just to viral transcription, but to the production of new infectious virions as well. And with that, I'd love to take any questions that you have, and I'd love to talk about this throughout the week. Thank you very much for listening.
Thank you very much, David. Any questions?
Thank you very much for the talk. Probably you said it and I missed it, but the reason why you are sure about the variations in the sequence is because you are making many copies of the singularized transcripts, and then you get the consensus of each one of the templates?
We actually have many cDNA copies from the beginning. From reverse transcription, we have cDNA copies, and each cDNA has its own barcode. So we can say that there were different mRNA transcripts present in that cell that share the same mutation, and that's how we generate the consensus sequences.
I'm thinking about the errors of the machine.
Yeah. So for one, the mutation rate of influenza virus is much, much higher than the mutation rate of the polymerase. So any errors are more likely to be from the influenza virus. But to account for mutation, especially during reverse transcription, we have unique barcodes added to every molecule that gets reverse transcribed. And so we use those cDNA barcodes to look for multiple copies of the same mutation in the same cell. So we have reasonably high confidence. It would be nice if we had deeper sequencing data, but we're at least usually looking at 2 out of 3 molecules in the cell carrying the mutation before we'll call it mutation in this data set. Yeah. Thank you.
Hi, sorry for the presentation. One quick question. What is MSG doing?
Yeah.
Why it provides advantage, or is it kind of count?
Yeah.
The others, or,
Yeah. Great question. If I can move my mouse back. I don't know if I can move my mouse back. Okay, so the function of this NS protein is basically to move viral genomes outside of the nucleus and into the cytoplasm so that the genome can be packaged into a new virus particle. And our theory is that when that is absent, you have many copies of viral genomes present in the nucleus, and those many copies of viral genomes are transcribed many times. And so our theory is that basically we were surprised. We thought high viral transcription would represent an advantage for the virus, but instead we think it represents the virus getting stuck in the nucleus and not able to exit.
Any other question? My question was connected to that. So it's not meaning that it's a higher transcription level, it just means probably the same rate, but it's just not being able to get capsulated. So it's just accumulated.
Exactly. That's my theory. Is this represents accumulation, not more production.
Yeah.
so do you have any information about the transcriptional state of the host cells?
Yes, we have the entire host transcriptome, and shockingly, we cannot detect any differences between cells with very high viral transcription and cells without. The host transcriptome is almost entirely unperturbed. There's no interferon. There's nothing that we could find. Although the data is available if you want to look in ways that we didn't.
Sorry, where the starting cells was like a cell line, or?
Yeah, clonal cell line, clonal virus.
Okay, so it might be that you pull up the primary cells.
Yeah, and that's work that's ongoing in the lab now.